Good evening, Professor. This is my personal timetable. In this week, I have to be continued the previous week work. The first one is the Corona Biosensor research paper that is deal with Prashant to for the characterization AFM, SEM, FTIR. Sir, we have contacted to Dongmin for helping the booking for these three different kind of characterization methods and instruments. So we found that KANC, KANC is available for doing this, and we have tried in Seoul National University and um, KIST and University of Seoul, but the cons is available for doing this, and we have to send our samples to the center, and they just uh, given us the results. So in the next week, we got the appointment, we got the chance to send the samples and for the characterization. And so once we got the result of all three characterizations, then we have to implement or to add on in the paper. So and we have to write the mentioned contents as well. So the 30% is remaining, 30 to 35% is remaining. But if I exclude the characterization part, so the 70% is already done, and simultaneously me, Prashant, and Jaya also work on the graphical abstract figure and also the uh, main figures also. So it about to finish in this week. And the second one is RF review paper. So in this case we got the rejection from the biotechnology advances due to having 20 I think 24% plagiarized content. So in this week, me and Prasant sit together to mm, do the remove the plagiarized content and to uh, set the our content on there. So in this week we have to do this, and then we have to finalize for the science journal, as you said to the Prashant, for the later content as well. And the task 3 is aptamer biosensor research. So in the last week, due to found the COVID things on the sixth floor, so the Ryunsang and Dongmin is not available and also is not present that time. So in this week, we have to recontinue this research. From tomorrow onwards, we continued this research using D3 and D5 chip, and because first of all I have to check which chip is better for this research. So that's why I have to take in two different kind of chips, and once we got the better results, so we move forward with that chip. And I have planned the recipe as well. So this thing, and the task four is PSA biosensor research. So we got the biosensor PSA antibody and antigen. So simultaneously also working on this, but due to having the last week problem, so I have to continue in this week as well. So I have to present my datas, my results, capacitive values in this week, at the end of this week, or in the next meeting PPT. And the third task 5 is AAO Biosensor research. So, as we discussed in the last meeting, our Ag protein particle is much higher and is not compatible with our existing AAO membrane. As per your suggestion, we have to be minimize the Ag particles. So we did the sonicator, and we also, after the sonicator, today Bruce did the LSPR measurement. So he will explain you in his PPT what we got the changes after the sonicator. And as per my understanding, the Ag particle will definitely turns into the minimizing level, minimizing size. So as I've discussed with the Bruce, so the Bruce said like the result is quite much different as per according to the previous results. So this is the new antibody data got it, and we move forward to using this too for with the graphene oxide. So it's already going on. So in this week I will present my data with the using of these two antibodies and antigen. So this is the research recipe, or we call research plan for the BSA. So we use here D5, and in this case we use graphene oxide as well, and so we have EDC and NHS as well. Now we got the antibody, and as per the different kind of concentration is here, I use 5 different kind of concentration. So according to the antigen concentration, immobilization on the antibody, and check the measurement at each step. If I didn't get any positive result at any step, so I have to stop it and think about how we can get the positive results, and if to tackle the to tackle the problems and to get the better results. So this is the AAO biosensor research work. So we have the AAO membrane that is having a two different kind of size, 75 nanometer or 95 nanometer pore size. So as per the last meeting, we did the sonicator, which is already done, and the Bruce did today the LSPR measurement. So he said like the result is quite different from the previous one. So maybe I think the sonicator is much more beneficial for us to get the smaller size of Ag particles. But the problem is we didn't put the Ag solution for a long time in the sonicator, because the sonicator itself having the temperature. It's, so that's why we just put only the half an hour or like 1 hour for the dissociation. So because the protein itself mixed with the Ag, so if we increase the temperature in the sonicator, the protein dissociate, so there is no worth for this machine. So we just put only half an hour or 1 hour for the dissociation process. So we did the LSPR measurement. So the Bruce will explain you in his PPT what we got in the LSPR measurement. So this is the recipe that we followed, and it's the same kind of recipe that we already use in my PSA, but the thing is we just use washing technique in each step with the PBS, and then we have to measure the LSPR measurement. And in this case, research plan 2, we just only skip the washing step and to continue the same process as we followed in the other research plans. So Bruce told me that he was thinking about this chemical because um, that chemical is already used in such several papers. So we already think about this, and so Bruce will contact to Dr. Daniel for book this chemical, and hopefully in this week they will order this chemical. So for SEM, we already present you the results. So the results is not quite good, but we have found the problem, like are where we going in the right direction or not. So that's why we have to first check with the SEM. So we found that the Ag protein complex is just passed through the AAO nanopore and is submerged. It's like uh, settle down onto the glass and to pass through the AO membrane, because Ag protein complex itself having the less particle size than the as compared to the AO, so that's why it just pass through the AO pore and just settle down onto the glass. And if I compare with this, so it's having a thick layer of the Ag protein in between the glass and the AO nanopore. But the AO nanopore size is similar as per the parameter from the AO company that we got. So the AO membrane is we found that is good, but why the Ag protein it comes pass through the AO membrane. So we found that here we use in this case we didn't use graphene oxide. We just simply put the Ag protein onto the surface, AO surface. But in this case, we mix with the AAO Ag protein and the graphene oxide, and then we immobilize onto the AO membrane. So the graphene itself having the large particles, so large flakes we call large flakes. So the large flakes mix with the Ag protein. So the circle part, if you see here, the circle part is the Ag protein, and the flake itself having the graphene. This flakes and this big flakes is a graphene oxide. So because of the graphene oxide, it does not penetrate into the AAO pore, and it just simply immobilized on the AO nanopore surface. So the graphene act like a barrier. Graphene act like a barrier to interact with the AO nanopore. So that's why it looks like this, and it just simply called as a very thick layer. So it is not good. So that's why we did the sonicator for the graphene oxide case also. So hopefully we got the small flakes, and then we mix together, and then we put onto the AO and check the LSPR or SEM. So hopefully we got a better result as per compared with this SEM results. So as I said, like this graphene flakes act like a barrier onto the AO pore because the graphene flakes itself having the large flake area. So that's why the Ag does not attach or does not penetrate into the AO pore. So that's why we got this image. So it's not good. It's not the good picture. It's not the good results as well. So we found that the particle size may differ the this research. So we did the sonicator and to we dissociate the particles of Ag nanoparticles and graphene oxide. So hopefully we will get the better results uh, for the next case. So it's already explained to you in the last meeting. So if there is a gap and also so we work on it for the sonicator, and this one too is so uh, the conclusion is we did the sonicator and we found that the Ag particles now we got the less size as compared to the previous one, and also I did the graphene oxide sonicate put into the sonicator for getting the small flakes. So if we have the small molecules, small particle size with the existing AO membrane, so now we will get the better results, and it's we will do the tremendous things to be done with this. And me and Bruce already discussed today's for the new things. So hopefully we will get the better results in the upcoming weeks, and will show you in the next meeting. Thank you, sir.